In this lecture, we will learn about transcription. Transcription is the second step of central dogma where DNA strand will act as a template to synthesize an RNA molecule. In this lecture, we will focus on bacterial transcription. Before we start talking about transcription, let us first focus on the central molecule in transcription, which is the RNA molecule. We previously learned about the structure of RNA. The monomeric unit for RNA are ribonucleotides. In the second prime carbon position in ribose, you have an OH group. If you remember DNA in deoxyribose, the second prime carbon position actually contain a hydrogen group. Okay, so RNA contain nitrogenous bases such as purines, including adenine and guanine, and also we have pyrimidines such as cytosine and uracil. Recalling, in DNA, we do not have a uracil. Instead of uracil, we have thiamine. Primary structure of RNA is a single strand, but RNA can have many different kinds of secondary structures. This is due to the complementary base pairing that can happen within that single-stranded RNA molecule. Let us quickly compare the structure of DNA and RNA. Both DNA and RNA are made of nucleotides. While ribonucleotides contain ribose sugar, deoxyribonucleotides contain deoxyribose sugar. This variation is due to the fact that I mentioned just now the 2' carbon position containing that OH group in ribose and hydrogen in deoxyribose. This variation also makes RNA more unstable and easily degraded. Both types of nucleotides are joined via phosphodiester bonds, and RNA, like I mentioned earlier, can have many secondary structures, while DNA is just a double helical structure. Even though these variations in RNA and DNA may look subtle, there are enzymes and proteins that specifically recognize DNA from RNA and specifically reacts with these two molecules. Let's refresh on the concept of central dogma. The central dogma actually illustrates on the flow of genetic information from DNA to protein. Information in DNA is first copied into RNA in a process called transcription, while RNA is then translated to an amino acid sequence of a protein. So now some viruses may seem to be not going in the direction of molecular flow. They can actually go backward. So this is because some viruses can have this enzyme called reverse transcriptase, which is an enzyme that allows them to convert RNA into DNA. In this lecture, we will learn about the second step of central dogma, which is transcription. Transcription is a process in which RNA polymerase will copy the information in DNA into an RNA molecule. Transcription in eukaryotes is much more complex compared to prokaryotes. So in this lecture, we will focus mostly in prokaryotic transcription. At a time, only a small portion of DNA will be transcribed. This is because transcription needs to happen only in 1 or 2 genes. So RNA polymerase does not have to completely synthesize a complete total RNA molecule that matches the total length of the DNA. So only a small portion of DNA will be transcribed into RNA. Also, when transcription happens, only one strand of DNA will be used at a time, and it is important to notice that in eukaryotes, only the regions of the chromosomes that compose of euchromatins are actually transcribable. DNA that are located in heterochromatin regions in chromosomes are non-transcribable. The process of transcription is carried out by this enzyme called RNA polymerase. And RNA polymerase actually requires a DNA template, raw material. This raw material actually includes nucleotides. So the nucleotides that is necessary for the synthesis of an RNA molecules are ribonucleoside triphosphates. Also, RNA polymerase requires a transcription apparatus that contain other proteins that are helping in transcription. So now the next task is for us to understand what is the coding region and what is the template strand, or coding strand versus the template strand. Genes are located in both strands of DNA, but at a given time only one DNA strand will be used as the template. The other strand is actually known as the non-template or the coding strand. The base sequence of the coding strand is similar to the base sequence of the primary RNA transcript, except the thymine is replaced with uracil in RNA. If you know the sequence of the coding strand, and you can easily get an idea of the sequence of the primary transcript, only you have to just change the thymine into uracil to indicate that it is a RNA transcript. Okay. Which strand of the DNA will be used for the transcription is actually determined by specific sequences that are located in DNA called promoters. So RNA polymerase, as similar to DNA polymerase, will synthesize RNA in the direction of 5' to 3'. Hence, the template strand has to be in the direction of 3' to 5'. If you look in this picture, you can see that this region is called a replication bubble. So in the blue color, you can find your RNA polymerase that is synthesizing in the direction of 5' to 3'. So your RNA transcript is given here as well. And if you look at the sequence in the DNA template and the coding strand, the nucleotide sequence is matching to the coding strand. Only you can see the thymines are replaced with uracil. That is why the same thing is indicated here. You can see the template strand. So RNA polymerase will use the template strand to make the complementary base pairing in the RNA transcript. When the RNA transcript is generated, the coding strand has the same sequence of the RNA transcript, except the thymines are actually replaced with uracils. So as mentioned in the previous slide, transcription can use only one DNA strand as the template at a time. Which DNA strand that is used is determined by other sequences available in DNA. A transcriptional unit is a stretch of DNA that encodes an RNA molecule and the sequences necessary for its transcription. Therefore, a transcriptional unit contains a promoter, which is a DNA sequence that is located in the upstream of the transcription start site, and this sequence will determine the template strand and the start site for the transcription. Also, in a transcriptional unit, you can find the RNA coding region. In this is the sequence of DNA that contain information that will be copied into RNA. Last but not least, you will have the terminator sequence that will signal the end of transcription. At a given time, when RNA is generated from the transcriptional unit, the promoter will not get transcribed, but the terminator will get transcribed into the primary RNA transcript. When you are looking at the transcriptional unit as indicated here, you can find these sequences we just talked about. We have the promoter that is located upstream of the transcription site, and then the RNA coding region and the transcription termination site at the downstream of the transcription initiation site. So any sequences that are above or like coming before the transcriptional start site is called upstream sequences, and any sequences coming after the transcriptional start site are called downstream sequences. And you can also recognize the two DNA strands here. One will be the template strand, the other will be the non-template strand, or you can also call the coding strand. So these are important to notice the numbering system for the transcriptional unit. So we give these plus and minus minuses for the transcriptional unit. So the plus mark actually you can find the plus marks where you have the DNA sequences where the transcription begin. So the first nucleotide where the transcription start is demarcated with a +1, and any sequences that or base pairs, DNA base pairs that comes after the start site will have +2, +3 like that. And sequences before the start sites are demarcated with a negative number. So for an example, if you have the promoter and you are looking at a specific number of a nucleotide in the promoter, we can mention it as -10. -10 means that 10 nucleotides before the +1 site, that is -10, and you can also mention -35, which is that 35 nucleotides before the transcription start site. So the sequences after the transcription site are positive, and then there are no zeros in the numbering system of the transcription unit. Now let's talk a little bit about RNA polymerase. RNA polymerase actually utilizes ribonucleoside triphosphates to catalyze the reaction and generate phosphodiester bonds. The energy for this reaction comes from the nucleoside triphosphate itself, where the two terminal phosphates are actually held in this high energy bond. When you break these two phosphate groups at the end of this triphosphate, nucleoside triphosphate, it will provide sufficient energy for the reaction catalyzed by RNA polymerase. Again, RNA polymerase is just similar to DNA polymerase. It polymerizes, adding new nucleotides in the direction of 5' to 3' direction. Bacterial transcription. So we are going to majorly focus on bacterial transcription in this lecture. So bacterial transcription is actually subdivided into 3 main steps to make it easy to study. The first step is called initiation. In this step, RNA polymerase will recognize the promoter and position itself on the DNA template. The second step is called elongation. In this step, RNA polymerase will synthesize RNA in 5' to 3' direction utilizing the DNA template for the complementary base pairing. Lastly, the last step is called termination. In termination, RNA polymerase will reach the terminator sequences that is located in DNA and dissociate from DNA and completes the transcription. We are going to go one by one for each of these steps, and we are going to discuss how each of what other important factors are happening in each of these steps. So let's talk a little bit about bacterial RNA polymerase. Bacteria, unlike eukaryotes, contain only one type of RNA polymerase, while eukaryotes, on the other hand, can have 3 different RNA polymerases, and each RNA polymerase can synthesize different type of RNA. Bacterial RNA polymerase have 5 different subunits. Namely, these include beta, beta dash, sigma, lambda, and omega. all 5 subunits together called the RNA holoenzyme. But the sigma subunit of the bacterial RNA polymerase can play a crucial role where this sigma subunit itself can recognize the promoter sequence that is located in DNA. Sigma subunit actually separate from the rest of the RNA polymerase, and it will scan the DNA to locate the promoter. RNA polymerase without the sigma subunit is actually called the core enzyme. So core enzyme alone can synthesize RNA. During initiation of transcription, several important steps happen. In summary, these includes recognition of the promoter, formation of the transcription bubble. RNA polymerase has the ability to unwind DNA and synthesize RNA in a small bubble that provides the single-stranded DNA template for the transcription. RNA polymerase is also capable of creating the first phosphodiester bond between two RNTPase. After the first bond is generated, the transcription initiation is completed, and transcription apparatus then escapes the promoter region, so the elongation can happen. So initiation actually include all 4 of these steps. So let's a little bit look in detail how the initiation process happen in bacteria. The recognition of the promoter is uttermost important for the transcription, as it provides the location of the gene or operon for the transcription. In bacteria, there are several consensus sequences in the upstream of the transcription, the transcription start site called -10 and -35 regions. Both of these regions are actually very important and can serve as promoter regions for bacterial RNA polymerase sigma factor. So sigma factor can locate both of these consensus sequences that are located in -10 and -35 regions. The consensus sequences are called consensus because in these sequences are actually conserved in many bacteria. During initiation, once the sigma factor binds to the promoter, RNA core enzyme is actually loaded into the template of DNA, and DNA started unwinding and transcription is actually starting. So the RNA polymerase is actually positioned over the transcription start site, which is the position +1, and now RNA polymerase can start adding nucleotides and transcription begins. As you note already, unlike DNA polymerase, RNA polymerase do not require a primer for adding the first nucleotide. So since RNA polymerase is capable of adding 2 RNTPase together without a primer, after the first nucleotide is added to the chain, the sigma factor actually get released from the RNA polymerase, and the core enzyme can catalyze to add the next nucleotide to the 3' OH group of the previous nucleotide. Once it is added, the RNA polymerase move downstream to the start site. So the end of the initiation actually demarcated by adding that first nucleotides together and leaving the promoter region and starting to go elongation step. In the elongation step, RNA polymerase moves downstream from the start site and elongate the RNA transcript. RNA polymerase progressively unwinds the DNA, and this creates a transcription bubble. It is about 18 nucleotide in length, where single-stranded DNA is exposed, and RNA polymerase can utilize that template of DNA to synthesize RNA. As RNA polymerase move downstream, DNA is unwounded, and behind of that replication bubble, DNA is wounded back. So the last step of bacterial RNA transcription is called termination. Termination of transcription is also very important, as this allow to demarcate the end of the transcription. So there are terminator sequences that are located in the transcriptional unit. These are DNA sequences. In bacteria, you can find two different methods for transcription termination. One is called row-dependent termination, where row protein is involved. The other method is called row-independent termination, which does not require a row protein. Let's first look at the row-dependent termination. In row-dependent termination, there are two DNA sequences that are important. The one is the terminator sequence that is located in DNA that signals the end of the transcription. And the other one is called row utilization site, or the RUT site, that is located in the RNA transcript. This RUT site is actually recognized by this protein called row factor. Row factor will actually bind to the RUT site and it start moving along the RNA transcript towards the 3' end of the RNA transcript. As it moves towards the 3' end of the RNA transcript, RNA polymerase will already catch up with the terminator sequence. So when the RNA polymerase catch up with the terminator sequence, it actually pauses. When it pauses, the row factor can catch up with that terminator sequence. When the row factor reaches the RNA-DNA hybrid, it can recognize the RNA-DNA hybrid and utilizes its capability of helicase activity to unwind RNA transcript from the DNA, and this actually demarcates the end of the transcription. And the other method of bacterial transcription termination is called row-independent termination. In row-independent termination, there are inverted repeats that are present in the terminator region in the DNA, as you can see here. Right next to these two inverted repeats, you can find a stretch of adenine nucleotides that are located right after these inverted repeats in the terminator region. So these inverted repeats will create a hair loop structure in RNA. This hairpin structure in RNA transcript can actually destabilize the AU pairing in DNA and RNA hybrid. So as you can see, when the RNA transcript creates that hairpin structure, RNA polymerase will still synthesize RNA and create a stretch of U's, as this is your RNA and DNA hybrid, which has a stretch of U and A. This hairpin structure can actually destabilize this AU pairing in the DNA and RNA hybrid. Then DNA and RNA hybrid will dissociate, which demarcates the end of the transcription. It is also worthwhile to notice that RNA transcript include the sequence for the terminator. As you can see, as it ends, this U pairs, the stretch of U's are also included in the primary RNA transcript. But the promoter region do not get transcribed into the RNA transcript. So that is the end of this lecture. So I hope you enjoy the lecture. So let us meet with another lecture that focuses on the eukaryotic transcription.